20 years ago, mankind made a colossal leap by charting the human genome. Today, scientists and doctors use this information to help patients understand inherited complex diseases and, in turn, take preventative measures to treat and abate different genetically caused diseases. Two major sources of clinical variant information are ClinVar, which is an NCBI hosted database, and the Leiden Open Variant Databases, or LOVD, which are several clinical genetic databases hosted in Europe. Many clinical groups in the United States tend only to use ClinVar because it is more well known. However, these groups overlook large amounts of relevant information because they are not aware it exists. This visualization sets out to communicate not only the existence of the LOVD platform, but also its utility to clinicians and leaders in the genetic testing industry.
The visualizations we've created to demonstrate the data include two stacked bar charts which show the percentage and count overlap of the gene variants between the databases, as well as a line graph which shows the different genes present in the database and how frequently those are classified into the different pathogenicity classifications. Next, we also have the scatter plot which shows the specific gene and the variants of that gene with the distance and frequency on the different axes. These variants can be selected to show further information on the right-hand side and hovered over to see the gene variant. We also have the gene structure showing exons, introns, and untranslated regions of the genome. And finally, a stacked bar chart showing the gene variants which were invalid variants and the reason for those being invalid classifications. All of these charts are updated by either selecting genes from the dropdown here or by selecting from the different filters on the left-hand side, such as variant type, pathogenicity classification, and then selecting the redraw button.
Aside from showing that LOVD contains variants that are not in ClinVar, probably the most important thing from all of this would be the ability to see where variants are located within a gene. So select a gene, I'll choose ARSA, and you can see that there are variants throughout the entirety of this whole gene. However, if you are to filter this and look at only pathogenic and likely pathogenic variants and reset the scatter plot, you can see that all of the variants are only within a certain region of the gene, and this does not include the untranslated regions. So if you're working in a clinic, for instance, and you had a variant that was in an untranslated region, you could assume that it is not going to be pathogenic because none of the other reported variants within that region have previously been pathogenic. Thank you.